Section 13.5, we're almost at the end, is going to focus on B cells. Alright, so B cells, so I mentioned this in section 13.4, talking about the differences between the cellular third line of defense and the humoral immunity, the third line of defense. That humoral is going to be directed by or indicate the B cells. So, you have your objectives here. We're going to look through table 13.6, that figure that shows the different types of B cells that are produced in the activation process. We want to look at a detailed drawing of an antibody, and then the functions of the antibodies, and then how the immune response can actually be identified at what phase you're in for an immune response by what antibodies a lab test can identify. So here is table 13.5 or 13.6 that looks at B cell activation, where at the very top of the page there, or the very top of that table, you have the microbe coming in and finding the very specific the B cell that is specific for that molecule. And so it's going to bind to the B cell receptor, and then it's going to pull that microbe inside of it, which you see in step 2, and it starts to digest it, to process that antigen, and then present it on its surface again to a T helper cell. Alright, then the T helper cell and the B cell actually come into contact with each other through those surface proteins. The T cell gets activated by the T cell gets activated, yes, by the binding of that antigen, and the T cell is then going to secrete different cytokines that will then basically, come from this cell over and activate the B cell. So here in step 4, you have the activated B cell. And then at that point, we move into step 5 there, where that activated B cell can differentiate, become a different type of B cell, and the options are going to be a regulatory B cell, a memory B cell, and then a plasma cell. Those are the 3 options. And then you have clonal expansion, and clonal expansion means that those 3 options are just simply going to expand in number. So here you have a group of memory cells. The memory cells are simply going to hang out, right? They're alive, they're doing well, they don't die very often. And so once you get exposed to something, alright, this specific microbe back here, these memory B cells are specific to this guy, and they are simply going to hang out in the bone marrow, in the lymph nodes, in the spleen, until this little guy gets back into your body. And then once it gets back into your body, the B cells don't have to go through this entire process, because you already have that downstream effect that was established the first time your body saw this microbe. So then, all of these memory cells can then react very quickly and bind specifically to this microbe and get rid of it before there's a problem. On this side, you have the plasma cells. And the plasma cells look very different from the other 2 types of cells. And that's because the plasma cells, their job is simply to produce antibody. Alright, if you remember, antibody is immunoglobulin that is secreted from the cell, so it's just floating around in the blood, in the lymph, and it is going to be there at actually pretty high numbers, so that if and when this microbe comes back, alright, microbe 1 down here, microbe 1 down here, or up here, it's the same guy, that then the antibody can bind to the microbe. And you can't see it very well here, but in your textbook, when the figure is blown up as big as it can be, there's a very specific interaction between that antibody and the microbe. and then the microbe is neutralized, which we'll see on the next slide, or on the next slide. So this slide is just going to look at the structure of the antibody. All right, the antibody, and this is an immunoglobulin in general. The immunoglobulin, whether it's a B cell receptor on the surface of a B cell or a secreted antibody, is going to have the same structure. And I started to talk about this earlier, but this is a better picture to try to describe what's going on. The antibody structure. 2 heavy chains. All right, this is a heavy chain, and it's because it's the biggest. All right, the 2 heavy chains are connected with each other with disulfide bonds, and it basically creates a Y. All right, then here you have the 2 light chains. One light chain here, it's lighter, so it's smaller, and then the other light chain down here. And together, those 4 protein strands make up the antibody. And this is a crystallization rendition of the antibody. All right, then up at the top of the antibody, or the Y, you have this area called the variable region. Variable means it's going to vary in terms of all of the different amino acids that are in here, and each dot or spot, corner or groove of this antibody that you see here would represent a different amino acid. There's a great variety of different amino acids that will come in this area and form the antigen binding site. You see kind of this hole right here. That's where the antigen is actually going to bind. But it does that in the antigen binding site on the variable portion of the antibody. And then this darker portion, the red maroon purple portion, is going to be the constant region. All right, and that's indicated by the C. All right, the FC, the constant fragment of the antibody. All right, so then that's going to be constant, but the variable region is variable, so it can bind to a lot of different antigens. All right, now looking at the function of the antibody. So now that you've seen the antibody, how it's created, and it's antibody, which means it's free-floating, there are 5 different functions of the antibodies. All right, one, it can neutralize an antibody. It can neutralize something. So here you have neutralization of this virus. All right, the virus has different ligands on its surface, different protein spikes, and it's going to use those protein spikes to bind to a receptor on a host cell. But if those ligands are covered up by the antibody, that virus is neutralized. It cannot get into a host cell. So that's neutralization. All right, this picture down here is agglutination. All right, agglutination is just agglutinating everything, all right, making a big clump of stuff. The antibody has 2 binding sites, so you can see how this one antibody is connecting or being a bridge between 2 bacteria cells, and that's because it has 2 antigen binding sites, and they're identical. So the antibody is going to find epitopes of the same amino acids on this bacteria cell and this bacteria cell. Then you have this antibody binding these 2 cells together, and this one, these 2 cells. So you get this clumping. That clumping helps to keep the bacteria from spreading. It can also, in a way, inhibit or impede the process of binary fission, all right, growth of the bacteria. And then there is opsonization. Opsonization is diagrammed here, where the antibody simply binds the bacteria's surface. All right, again, you're seeing it attached at both antigen binding sites here. And then if you remember, this bottom part of the antibody is the FC portion, that constant portion, while your macrophage has different receptors that are going to specifically bind to that FC portion, and it's going to help facilitate the engulfment or the engulfment by being able to specifically bind that Fc portion of the antibody with the receptor on a phagocyte. All right, this being the macrophage. So the antibody acts as an opsonin. All right, that opsonin is attached to the bacteria. That helps the phagocyte to ingest it more efficiently. All right, then you can lyse the cell by activating complement, and that's what this portion of the figure shows, where you see this little hole here, and the material from the cell is moving out into the environment. And what happens is the antibody binds to the cell surface, and then there are signals that cause a group of serum proteins to come and attach to the bacteria cell because that antibody is there, and the proteins build a channel, or basically a hole, in which material moves the cell. And so then that cell is going to break open because of the presence of all those complement proteins. And then antitoxin. All right, very similar to neutralization, but the antibody is specific for a toxin that is secreted from the bacteria cell. So diseases such as tetanus. All right, tetanus is an infection, yes, but the problem that causes death is actually the toxin that is secreted by the bacteria cell once it gets inside of you. And so the vaccine is actually for the toxin, not actually for the bacteria. And the antibody will bind to the toxin and neutralize it by being an antitoxin. All right, then here we have a picture of the immune response. And what we didn't talk about, because there's just a lot of information here, is that there are different types of antibodies. And the two major forms for just a normal run-of-the-mill immune response are going to be IgM and IgG. And the lines that you have here, you have this legend here where the red line here is actually IgM, and for some reason that little red bar is not there. The dark blue line is the combined or total amount of Ig, immunoglobulin, and then the light blue is IgG. So here on the y-axis you have the antibody titer, how much antibody is actually in you, and that is described on the next slide. And then you have time on the x-axis. So you have an exposure, the very first exposure to an antigen. This could be real life, it could be through a vaccine. And it takes some time where your immune system is trying to figure things out. And then after 7 to 10 days, you have this little jump in IgM levels, and it falls off. But at the same time, your B cells are still continuing to see that antigen and to further differentiate. And so the IgM B cell will actually eventually differentiate into an IgG, or some of those. And so IgG comes after IgM, and it's a little higher. And then again, you have this blue line, which is the combined number of both the IgM and the IgG. And so this here is the primary immune response, the first immune response. Then you have however long interval time, and you have a second exposure. And the difference between the first and the second, or there's a couple, but a major one, is you don't have this latent period. You don't have this lag time where your immune system isn't doing anything, right? But rather almost immediately your immunoglobulin numbers increase. But this time IgM is still a little slow, doesn't produce a whole lot, not actually maybe even less than the primary immune response. But IgG jumps up quickly, immediately and fast, and it stays up high. So you can have your antibodies titered to see how much of a specific antibody you have, whether it's IgM or IgG. That's very interesting to do, but you can, by doing that, you can determine, well, is the person in the primary immune response, are they in the secondary immune response, are they way out towards the end where that infection may actually be coming to an end. All right, and then this is just some of that, some